Take notes, stop me if you don't understand something, and but most importantly, hold on, because it's going to go quickly. I'll summarize things for you that you should know, and believe me, I don't expect you to know everything about the immune system in 2 or 3 lectures, because it ain't possible. So, the other thing is that clickers are not working right now because the university has this dual authentication system thing. Do you know what I'm talking about? Yeah. So Turning Technologies can't deal with that. So ASU and Turning Technologies. So everybody's going to get 2 points. Even people that aren't here are going to get 2 points, 2 free clicker points. But you can use your hands. So the first thing is, how much do you know about the immune system? So how many people, we already said how many people. A, you took an immunology course. We had people show up their hands once again. You took an immunology course. Okay. Few, even less than last time, it's interesting. So B and Lou, we have some immunology in another course. Awesome. So do you know what B cells are and what T cells are?
Yeah.
Okay, well that makes my job a little bit easier. Antibodies, antigens, stuff like that, so you kind of know what that is, which is good. Can everybody hear me? Because I know you've had some problems. Or D, what's the immune system? Okay. Or E, my grandfather was Ralph Steinman. Nobody knew. So who is Ralph Steinman, right? Well, Ralph Steinman won the Nobel Prize for discovering a special kind of antigen-presenting cell. His work with a special kind of antigen-presenting cell called dendritic cells. Dendritic cells are really, really good antigen-presenting cells, and they're so important because dendritic cells form the interface between the adaptive immune system and the innate immune system. So dendritic cells are part of the innate immune system, but they're really at the interface. And we'll be talking a little bit about dendritic cells and macrophages. And hopefully, if you took some immunology before, you know what a macrophage is. How many people know what a macrophage is? Okay, so most people. So macrophages are good at phagocytosis. Okay, so the first question is, why do we have an immune system? Anybody got any clues? Why do we have an immune system? Protect us from pathogens, right? Okay, so that's one question. Another answer. That's one answer. Another question was keep us alive. Any other answers? Okay. So, to protect us from microbial pathogens. That's the right answer here. So here's a harder question. What is the function of the immune system? And they're not the same answer. Anybody got any ideas? So you're close. That's pretty close. There's a lot of homeostatic mechanisms in the body, in mammals, as well as in any organism, a lot of homeostatic systems. Anybody want to expand on that?
To develop antibodies.
To develop antibodies? Okay, so that's one function of the immune system. So if we kind of put all these 3 answers together, we get toward what we're looking for, is to differentiate self from non-self. So early on, the immune system, when you just have a fetal immune system, and just right after you're born, your stem cells, and we'll talk about stem cells in a second, from your bone marrow decide whether or not they're going to become T cells or not. And they actually kind of get educated on what self is. So your immune system knows what self is, and everything else, as far as it's concerned, is non-self. So if you think about it, that means my immune system knows what I look like, but it really doesn't know what your proteins look like. And in most cases, our proteins are pretty much the same. But the immune system, because we know that I can't give you a kidney and you can't give me a liver or a lung or something like that without me rejecting it. So there's some very important molecules that the immune system uses to not only present antigens, but those are also the same molecules that it uses to be involved in tissue rejection. Okay, and we'll get to some of that. We won't talk about tissue rejection, MHC molecules involved in tissue rejection, because that's a deeper immunology subject. But those molecules, specifically major histocompatibility complex molecules, are involved in presenting antigen. Okay, and then we have to present antigen to the T cells because we have to know if we're infected with something. Okay, so I will try to give objectives with all my lectures, because I think if you read the objectives, people always say, well, how do I study? This is so much material, it's so hard, how do I study? Well, if you read the objectives and you know something around the objectives, then you should do okay on the exam. And if you study my PowerPoints, and if you don't understand what's in the PowerPoints, and then you read the textbook, then you'll do okay on the exam. And then if you're still having problems, either ask the TA or ask me. Okay, so the first thing we'll talk about today is the difference between infection and disease, right? Explain how a phagocytic cell recognizes a microbe. We'll talk about that today. We'll talk about inflammation. We'll talk about the names and functions of innate immune cells. We'll probably talk about this first. And then we'll talk a little bit about complement. Okay, how many people have talked about complement in other courses? Or have been in other courses where I talked about complement? Sometimes I say things backwards. Okay, so now we'll talk about, today we'll talk about innate immunity. And the rest of, after today, we'll talk about adaptive immunity. We have to talk about innate immunity because it's been underappreciated for a long time, and it's really important because when you get that first virus or the first bacteria, and remember, bacteria are everywhere, the innate immune system is really, really important. As a matter of fact, it's present at birth, and things that we don't even really think about are really and truly part of your innate immunity. And those are physical or chemical barriers to infection, right? So they're kind of non-specific responses that would destroy invading pathogens. And you might be thinking, well, something I know that's non-specific is... So what's something that's non-specific that might destroy an invading pathogen? Skin? Absolutely. Mucus? Yeah, they get trapped in mucus. Did you have lunch? Remember, there's lots of bacteria in whatever you ate for lunch, whether you know it or like it or want to think about it or not.
Your stomach acid?
Yeah, your stomach acid, right, absolutely. So those are things that are part of the innate immune system. And then we'll talk about adaptive immunity next time, and that's a reaction to specific antigens. And what you really need to know about adaptive immunity is it's antibody-driven and T-cell driven. The specificity develops as the need arises, and it retains immunological memory. So when I talk about adaptive immunity, I'm going to be talking about immunological memory. And if I ask you a question on an exam, what's the difference between innate and adaptive immunity, you're going to think about, well, adaptive immunity has immunological memory. I know that that's B cells and T cells, and not antigen-presenting cells or phagocytic cells. Okay, so we already said that part of the innate host defense is skin. So what's in skin? So there's keratinocytes, right? There's oil, so bacteria kind of get trapped in oil, and they don't like that. There's also just below your your stratum corneum in your skin, there are these cells called Langerhans cells. And Langerhans cells are skin-associated dendritic cells. Remember, Ralph Steinman discovered dendritic cells and did work with dendritic cells. He's not your grandfather. But Langerhans cells are dendritic cells, and we just call them Langerhans cells because the guy discovered them. His name is Langerhans. But they're dendritic cells in skin. So skin is a very, very active, immunologically active organ. Very, because it has to be, right? Bacteria are everywhere, and we cut ourselves and wipe our hands on the counter, things like that. So mucus, we also talked about mucus, right? So mucus traps the pathogens, and once the pathogens are trapped in mucus, there's mucolytic, or there's enzymes in mucus that destroy bacteria. Sometimes mucus gets too crowded with bacteria, so we have to use mucolytics to break up the mucus. That's a problem with patients that have cystic fibrosis disease, so they get too much bacteria in the mucus, and they actually have to break up the mucus. We can cough up mucus, right, and then swallow it, and we have stomach acid that takes care of the bacteria that are in the mucus that we swallow. And then cilia, of course, remove microbes even before they get to the lungs. The lungs are, even though we breathe in air, the lungs are semi-sterile unless you get a lung infection like TB or valley fever. Okay, so there's also chemical and protein methods of innate immunity. So lysozyme is an enzyme where does anybody know where lysozyme is?
Tears.
In tears, absolutely. So if no one got it, I was going to say, I'll cry. That was going to be a good hint. If you don't get this answer, then that would be a good. But I didn't get to say that because you knew right away that lysozyme was in tears. Okay, so stomach acid we already went over. Phospholipases, these are things that break up bacterial cell walls, bacterial components. Proteases, so a protease is something that cleaves up proteins, right? So we have specific antibacterial proteases. RNases, so some viruses are made of RNA, and we'll be learning about those soon. And RNases cleave up that RNA and those viruses. Okay, so here's what lysozyme does. Here's a N-acetylglucosamine and acetyluramic acid. Lysozyme actually cleaves this bond here between NAG and NAM. Okay, and if it cleaves that bond, what do you think it does to the peptidoglycan cell wall? Right, it breaks it, absolutely. So lysozyme actually breaks up bacterial cell walls. And some of you working in laboratories may do an experiment where you lyse bacteria, and sometimes you use lysozyme to lyse those bacteria because it breaks up the cell wall and then just leaves the cell membrane. And that's easy to destroy. Okay, so there's other molecules called defensins, and these are cationic peptides. So they're typically, when I say small, so a peptide is like a small protein, and the way proteins we say probably are larger than, I would say, 30 amino acids. So if somebody says it's a peptide, you know that it's smaller than 30 amino acids, and that's kind of a good figure to use. Proteins are kind of larger than 30 amino acids. So these are cationic peptides. So if they're cationic, what charge do they have?
Positive.
Positive, right, absolutely. Okay, so, and what they do is they insert into bacterial cell membranes, and they kind of make holes in it, make pores in it, and lots of different cells make these, keratinocytes, liver cells, etc. And then finally today we'll talk a little bit about complement, and what complement is, it's a cascade of proteins in blood that's produced by the liver. And what they do is they bind to the surface of bacteria and punch holes in the surfaces of bacteria. Okay? And that's what we'll be talking about, the alternative pathway of complement today. But next time we'll be talking about the classical pathway of complement. And the classical pathway of complement interacts with antibodies, so it interacts with the adaptive immune system. So I talk about antibodies, that's the adaptive immune system. Okay, so innate versus adaptive immunity, what do I need to know? So as far as the innate immunity goes, it's the response to the pathogen is the same. Okay, so macrophages are always going to engulf bacteria the same way. Okay, dendritic cells, same way. Okay, neutrophils are all going to do it the same way. But adaptive immunity, the response to the pathogen gets better. The first antibody response that you make to a virus or bacteria is not as good as later in infection when you're actually eliminating or neutralizing that virus or eliminating the bacteria. Okay, so it actually gets better. So adaptive immunity has immunological memory and it gets better. And it's composed of B and T cells. Okay, so the cells we're going to be concerned with for innate immunity are macrophages, dendritic cells. They do the same thing. Dendritic cells are just better. Natural killer cells. Okay, I'll talk a little. Let me skip these, but I'll talk a little bit about it if I have time. Neutrophils are really, really important. So does anybody know anybody who's had chemotherapy? That had cancer and chemotherapy? Right. So the first thing that happens when people get certain types of chemotherapy, not all chemotherapies, is that their neutrophils are killed. So their neutrophils are growing in bone marrow, and the neutrophils are killed because neutrophils are a very rapidly generating cell, and chemotherapy kills very rapidly generating cells because cancer grows, it's rapidly divided. Cancer cells are rapidly dividing. So neutrophil, if you don't have any neutrophils, you're prone to bacterial infection. So neutrophils are really, really good at phagocytosing bacteria. That's their primary job. Eosinophils are important in tissues, and they're primarily, kind of evolutionarily, they're very important for worm parasites. And since we don't really deal too much with worm parasites here in the United States and in first world countries, these are mainly problems in allergy now. So raise your hand if you have hay fever or allergies. My hand is up because I have it for sure. So, so you'll have you have more eosinophils and IgE on those eosinophils than people without allergies. Basophils are the same as eosinophils, but basophils are in blood. They're not in tissues. So eosinophils in tissues, basophils are in blood, primarily for worm parasites. Okay, so cells of the immune system, right? So we just kind of covered, so white blood cells, blood looks red, but you actually have lots of white blood cells, okay? And white blood cells are formed because stem cells in your bone marrow differentiate into all your different cells. T cells, B cells, which are lymphocytes, and in general, we call white blood cells leukocytes. Okay? So if somebody says white blood cell, you can correct them now. You can say, if your grandmother says white blood cell or your parents or something, you can say, no, that's a leukocyte. I learned that in microbes. So we have between 5 and 10,000 leukocytes per microliter, right? So a microliter is just that much volume, right? So it's 1,000 times less than a mil. But we have 5 to 10,000 leukocytes per microliter of blood. Half of those are neutrophils. So neutrophils are pretty important because there's lots of them. And like I said, if you don't have neutrophils, you'll get bacterial infections and they can be lethal. 25 to 40% of those 5 to 10,000 leukocytes are lymphocytes. Okay, so lymphocytes we're going to call B cells and T cells. Okay, so that's part of the adaptive immune system. So leukocytes in general, lymphocytes are B cells and T cells. And then 5% of those cells are monocytes, and monocytes are precursors to macrophages. So a monocyte is kind of a round cell floating around in the blood, and it doesn't become a macrophage until it hits tissue. Okay, so this would be either a B cell or a T cell. We have no idea. Probably not a neutrophil. Neutrophils, well, it could be a neutrophil, I guess. So this is a white blood cell. And they're about the same size, a little bit bigger than a red blood cell, except when they're activated. When they're activated, they'll get about 2 to 3 times the size of a red blood cell. Okay, so the remainder of these cells here are granulocytes. And when I say granulocytes, I mean basophils and eosinophils. And we can tell they're granulocytes because when we stain them, here I have a picture here. Yeah, when we stain them, these look like granules inside. Here's an eosinophil, here's a basophil. They look like granules inside the cell. Okay. And actually, when they're activated, sometimes they degranulate. And when they degranulate, like if they're near a worm parasite and they degranulate, then they kill the worm parasite, or they heavily damage it. Okay. All right, so hematopoiesis. You probably haven't heard the term hematopoiesis, and you don't use that term every day, unless you're an immunologist studying hematopoiesis. So this is the formation of blood cells. Okay, so you start out with a hematopoietic stem cell, or an HSC. You don't have to know that. You just have to know the concept of hematopoiesis. You start out with a stem cell. This is in your bone marrow. It doesn't know yet whether it's going to become a B cell, a T cell, a red cell, a neutrophil. It doesn't know. So it's a stem cell. Okay? These are the cells that we transplant to. If you're going to get a bone marrow transplant, these are the cells that we transplant into somebody. Okay? From a donor. And then you don't have to know all these things. So a stem cell goes down the pathway. This is a progenitor cell here. CLP here stands for common lymphoid progenitor. So, but very early on, cells decide whether they're going to become a B cell, a T cell, or an NK cell is actually a lymphoid cell as well. So they decide whether they're going to become lymphocytes or not. Okay, so this is, remember, the adaptive side. And then you have all these other cells here. Then you have a red cell. Here's a megakaryocyte, which eventually becomes platelets, neutrophils. Neutrophils are also called polys, or PMNs, polymorphonuclear leukocyte. And it's morphonuclear leukocyte because it has these multiple globules here in the nucleus. So anytime you're looking at a blood smear, has anybody ever looked at a blood smear before? Maybe in their job, something like that? Right. So you probably see some neutrophils. If you do a Rytgem stain, you'll see some neutrophils. And it has this multilobular nucleus. Okay. Monocytes, remember, they're round-looking cells. Sometimes they have a horseshoe-shaped nucleus. And as soon as they hit tissues, then they differentiate and they become a macrophage. And they get all these processes all over. Okay. What else? So sometimes what I'll do is I'll show you the picture, and I'll say everything that's on the next slide in text, so I'll just kind of skip over the text. Sometimes I'll review it if I think it's important. So just to review here, neutrophils and monocytes, they're the ones that phagocytose or engulf and destroy microbes. Just talked about monocytes differentiating into macrophages. You can also push monocytes to differentiate into dendritic cells, which are a special kind of dendritic cells, or a very effective monocyte. I already told you about the basophils and eosinophils degranulating, and they poison microbes like worms. And then lymphocytes, remember, are T cells, B cells. So T cells have a T cell receptor on their surface. B cells secrete antibodies. So T cells do not secrete a T cell receptor. They just have it on the surface. B cells will actually secrete an antibody. Okay? And then there's NK cells, and they're really kind of a special cell. And if we get to it today, we'll talk about them, but I may skip it. They kill tumor cells and virally infected cells, obviously. Okay, so now we're going to start to talk a little bit about phagocytosis. So we have a bacteria or even a virus, And what happens is, say you have bacteria in your blood for some reason, or in a tissue, and this is a phagocytic cell like a macrophage here, or a neutrophil. So these are neutrophils, right? A monocyte in blood, and here's a macrophage. See, it kind of has all these processes that can go and kind of lasso, like a cowboy. It can kind of lasso the bacteria. So here's the bacteria right here, and bacteria actually have things on their surfaces that will make them adhere, or it kind of makes them get caught by phagocytic cells like neutrophils or macrophages, and then the phagocytic cell will ingest the bacteria. Could be a virus too, by the way. And then once the bacteria is ingested, it forms a phagosome. Okay, so it's a phagocyte and forms a phagosome. And then what the phagosome does is it fuses with something called the lysosome. Okay, and the lysosome has proteases and special enzymes in it. And the proteases and other enzymes work at a low pH. So as soon as these two fuse together, we get kind of a low pH environment here, and these are acidic proteases, and as you can see, they break down the bacteria. So they start to destroy the bacteria. Okay? And this is a good figure up until about this point here, where it's destroying the microbe, and then I hate the figure, because this is not exactly what it does, and we'll get to another slide showing what a microbe does. I mean, maybe a neutrophil will do this, but neutrophils are boring because they just do the same thing. They just phagocytose bacteria all the time. Okay. So is everybody good with this level? Because we're going to dig a little bit deeper into phagocytosis. Everybody good with this level of phagocytosis? Fusion of a phagosome with a lysosome, activation of acidic proteases, breakdown of the bacteria, and then some other stuff happens, but part of it is released. All right, so the other thing in the innate immune system is that you have, remember, you ate lunch today, or some of you ate lunch today, and there's bacteria and everything, so you ate even live bacteria. There's a lot of dead bacteria there, but you ate live bacteria too. So the immune system has developed something called gut-associated lymphoid tissue, and within the gut-associated lymphoid tissues, there's these things called Peyer's patches. So this would be the small intestine here, and the Peyer's patches would be areas of lymphoid activity. So these are kind of like mini lymph nodes. So lymph nodes are areas of. Everybody know what a lymph node is? You've taken some anatomy. So a lymph node is like a little. It's a piece of tissue that's a station where T cells and B cells and phagocytic cells get together and kind of talk. It's a chat room for lymphocytes. That was a joke. So in the Peyer's patches, then, if you have bacteria, and we do have bacteria in our guts, and we don't exactly understand. There's a lot about immunology that we don't understand, but the bacteria in your gut get taken up by these vesicles and M cells, and then you have specialized dendritic cells that figure out which bacteria are the good bacteria in your gut and which bacteria are the bad bacteria in your gut, okay? Because we all know that we can eat some bad bacteria and get, bad diarrhea, something like that, or have a viral infection. But these phagocytic cells here figure out which is good and which are bad. So we don't actually kill our good bacteria. And there's bacterial reasons for that. There's a lot of hypotheses. There's bacterial reasons for that and host reasons for that as well. That's kind of for an immunology course. Okay, so these are M cells that take up these bacteria and sort things out, good versus bad. Okay, so now let's just watch watch a movie here. So get your popcorn out, put your feet up, and we'll watch a movie real quick. Basic inflammatory response. Oh, you know what? Before we do this, I got to hook up the microphone. And hopefully this... Okay. Let's do it this way.
What? You're not using it? I know. Are you gonna use one?
You have 3 on.
I know, but are you gonna use one?
You know what? We may actually skip this because this is on the Norton website. So maybe we'll skip this because I don't think I can get the sound to work because I don't have the sound projector or the like the iPod connector thing that goes into your computer. Oh, wait, here it is.
If it's captioned, everybody can read it.
Oh, okay. Hang on. So this is it?
That should be.
Uh-oh. I'm stuck.
Max. I noticed like a bunch of our teachers use like
Let's try this again.
MacBook Pros.
Your dream came true. Full recording.
You want me to try on my phone? Challenging me? Very challenging.
All right. Well, so I'll narrate this while it goes. Okay. So you're not going to have the benefit of the nice announcer, but you'll have the benefit of me, unfortunately. Okay. So this is skin here, and obviously this person has a splinter, right? So if you have a splinter, remember bacteria are everywhere, even on splinters. Let's just say this was lying in the dirt, so there's potentially some Clostridium bacteria here, so things that live in the dirt. And these bacteria have, the bacteria are always shedding things. They're very messy. And these other cells in the skin here, remember these are, what kind of cells are these in the skin? They're kind of like macrophages in the skin, but they're really good. So there's special dendritic cells in the skin called Langerhans cells, right? Okay, so these are Langerhans cells here in the skin, and let's see if I can make this go again. So now what's happening is they're just kind of patrolling around here, and as you notice, there's a, whoops, that's bad. So it's going to keep going. So if you notice here, there's neutrophils every once in a while, or phagocytic cells that come by, lots of red blood cells. These cells kind of patrol around in the skin, and they're going to contact these bacteria. Once they contact the bacteria, that's a signal for those cells to start secreting. See, they're phagocytizing them. Then they're going to start secreting things called cytokines. Okay, so those cytokines are very special because the cytokines, we'll talk about what kind of cytokines they are here in a second. What they're going to do is they're going to cause these endothelial cells to do something. They're kind of right now, these endothelial cells are really tight together, right? Because in a blood vessel, you don't want the endothelial cells that line the inside of your blood vessel to be loose, because if they're loose, then you really don't have a blood vessel anymore, right? And you get, so anybody ever had swelling if you cut your finger and you have swelling, right? And it's kind of squishy there, right? If it is kind of squishy, then what happens is that means that you have a lot of serum or plasma that's gone through this blood vessel here into your tissue. Okay, so that's part of the process of swelling. But these cytokines are gonna force these endothelial cells to open up a little bit, and they're going to allow these phagocytic cells out, like neutrophils. They're going to allow them out. Hang on. All right. So the first thing that's going to happen before it allows them out is the cytokines are going to cause selectins to be expressed on the surface of endothelial cells. And the selectins make the neutrophils slow down, okay? So they're racing through the blood vessel, and then they slow down as soon as they hit the selectins. And then once they hit those selectins, then it's going to cause integrins to be produced on the neutrophil, and then they're going to stick. So it's kind of like Velcro. So it kind of slows down, and eventually it sticks. Okay. Sometimes. Okay. So now it's stuck, right? So it's stuck here, and in a second, as soon as the guy gets talking about some other stuff that he's, so there's some bradykinin. We're not going to talk about bradykinin. That's, you don't have to know that. But these tight junctions between the endothelial cells open up a little bit, and then this neutrophil can extravasate. So that's kind of a big word. Extravasate through. And when it extravasates through, then it really does its job. Then it starts to eat all the bacteria here. And then what happens when you have lots of these things? You know, it only shows one of them happening here, right? So, yes, we get mast cells. We're not too worried about mast cells. When we get some histamine release, and that's going to make just get swelling here. So we have plasma released. That's swelling. You don't have to worry too much about that. But this is on the Norton website, so I would encourage you to watch this. You don't have to worry about prostaglandins either. Prostaglandins are produced, and they cause pain. Bradykinin also causes pain. And what do you take for pain? Right. So you take aspirin, you take a Tylenol, right? So those of this aspirin and Tylenol inhibit prostaglandin production. Okay. So, all right. So let's just kind of go through the steps a little once again. So the inflammatory process, because this is one of the things on your objectives that I want you to know is the inflammatory process. Okay, so you get an infection here, okay, and then as soon as the bacteria hit the phagocytic cells in the skin, cytokines are released. Those cytokines come down and they cause selectins to be expressed on endothelial cells. Okay, so we got selectins here on endothelial cells. The selectins are going to make the phagocytes slow down, slow, and then integrins are going to cause it to stop. So the integrins come up on the phagocyte, and they're going to cause it to stop. Once it stops, the cytokines are still helping the endothelial cells pull apart. They can extravasate through into the tissue where there's this gradient here of chemokines and bacterial products, and they swim up to the bacteria, and then they end up killing, endocytosing and killing these bacteria. And then there's actually a bunch of neutrophils here. And what are a bunch of neutrophils all together? There are a bunch of macrophages and neutrophils and everything that has phagocytosed that. It's kind of a gross term.
Pus.
Right? Pus, right, exactly. Pus is good. It sounds gross, but pus is actually good. Because that means you have an immune system, and that immune system has endocytosed and phagocytosed the bad bacteria. Okay. So here's a little bit more complicated figure, and this is in your textbook, but and you don't have to know about. So this here, people ask me, students have asked me in the past, well, what is this FMET-LuV? This is a peptide from the bacteria that will help activate the neutrophils. You don't really have to know that. You don't have to know anything about mast cells or histamine or prostaglandins or bradykinin. But I do want you to know that as the cytokines are produced by the phagocytic cells in the skin, they cause selectins to be expressed on the endothelial cells, and these selectins cause slowing and then slow rolling, and then the integrins are expressed on the surface of the neutrophils, and then the neutrophils stop, and then they extravasate in, and then they eat up the bacteria. Okay? So cytokines are things that are secreted by all leukocytes. Chemokines attract. So chemokines are kind of like a gradient. So you can think of lymphocytes as bloodhounds. So chemokines, there's just one molecule of chemokine, then there'll be 2 molecules, 3 molecules. So chemokines kind of attract lymphocytes toward a reaction. Okay? So lymphocytes are like bloodhounds. All right. The text on the next few slides are just what I talked about. So I'm kind of going to skip these. So we have bradykinin in here. I'm not going to test you on bradykinin. So what else do you know about histamine? Histamine, good or bad? So you get right. So you can get right. So histamine can be released when you have tissue damage. You can get a bee sting. But is anybody taking an antihistamine right now? I am. I had horrible allergies yesterday. I don't know why. Right. So an antihistamine prevents histamine from being released. So it's mainly an allergic. So histamine is released by mast cells and eosinophils and basophils, and that helps kill worm parasites. But it also causes problems for us that have allergies. Okay, so there's another phagocytosis movie here. What the heck? Let's go for it. Okay, remember we're going to talk about neutrophils, phagocytosis movie. Monocytes are also phagocytic. They become more phagocytic when they become macrophages, when they hit tissues. This is a macrophage. It looks like when it hits the tissue. Here it is, engulfing bacteria. I think there's a cartoon coming up. Yeah, the announcer here is talking about something that I'm not concerned with. Okay, so here's a macrophage, right? Here's a bacteria. So we've kind of already seen this. And there are little receptors on innate immune cells. We'll talk about these in a second. They're called toll-like receptors. So what's this right here? Phagosome, right. What's this? Lysosome. Are they going to fuse?
Yes.
Yeah. Okay, so now what's happening?
Acidic.
So the proteases are in there. They're breaking up the bacteria. Okay. The announcer is saying something else that's worthless. There are acidic proteases, probably DNases in there as well. Okay, so now here's something important. Let's stop. My favorite molecule. Okay. So, since this is a regular, normal cell in your body, it's a macrophage. You have this cruising around your tissues and in your blood as monocytes. It's making what all cells do. It's called an MHC molecule. Okay, so MHC molecule, the job of an MHC molecule is to present peptide. Remember, what are peptides? Yeah, so they're short proteins. And the peptides, the size of the peptides that MHC presents are either about 9 amino acids, sometimes they're 14 amino acids, depending on the type of MHC it is. But But the reason this is in this picture is because the MHC is made normally by the cell. It's made whether the cell is infected or not. It's always made by the cell. So we call this MHC, major histocompatibility complex. Histo means tissue. And compatibility means that if you have a different MHC than I have a different MHC, that means we're not compatible. So most of us in here probably share a few, but mostly we're not compatible, which is why organ transplants and bone marrow transplants are so difficult, because we recognize that we only know what our self-MHC is. We don't know what somebody else's MHC is, and we consider that as foreign. So we know what our self-MHC is. The T cells know what our self-MHC is. Okay, so we have these MHCs, and there are gene families. It's not just one. It's not simple like rocket science. It's immunology. So this MHC is being made, it's being made by the ER. It's glycosylated in the Golgi. It's going to go out to the surface, but before it goes out to the surface, it's going to collect a foreign peptide. And the reason it's going to collect a foreign peptide, so when I say foreign, is this bacteria is pathogenic. Let's just pretend the pink bacteria is pathogenic. And this foreign peptide here is from the bacteria. Okay? So the host is going, the T cells are going to come in here. By the way, T cells recognize MHC molecules. Immunology is a big circle and it's hard to break in. So I'm going to introduce new terms kind of slowly. So T cells are going to recognize this peptide plus MHC, and we'll talk about that either in a couple of lectures. Maybe one lecture from now. How many immunology lectures am I giving? 2 or 3? Just 2? I have to look at the syllabus and see. Okay. When do we finish class? 1:15 or 1:30? Oh, good. I have lots of time. I just got off the bus. I really don't know what's going on. Yes, ma'am.
You said it presents foreign peptides to
T cells.
T cells.
T cells. Since most of you have taken immunology in here, you know what a T cell is. A T cell is part of the adaptive immune response. Okay. Let's try to keep going here. All right, here's the good part. Right, so a foreign peptide complexes with this MHC molecule and gets put on the surface. So now, the T cell is going to know, how do I stop this? Macrophages or antigen? Okay. So this process normally happens. So we have MHC molecules made all the time, and if a macrophage is not infected, it'll just take one of our own peptides and put it on the surface, and then T cells will come in and they'll give it a little kiss and they're there and say, I know you, I love you, you're your own cell, and then it'll just go off, right? But remember, I said the function of the immune system, remember, is to recognize, what did I say? The function of the immune system is what?
Between self and non-self.
Self and non-self. Thank you. Sometimes I get confused. So self and non-self. So is this bacterial peptide self or non-self? Non-self, absolutely. So T cells know what self is. If this were just a regular, like an insulin-derived peptide, something like that, it would just go up here and say, yeah, I know you, that's cool. And then, but this is a bacterial peptide, and a T cell would come up here with its T cell receptor and say, whoa, this is different. And it would start to get activated. We'll talk about T cell activation next time. But a T cell would get very excited that something bad is going on here. Okay? So that's kind of the important point that I want to get across here, is that this macrophage, or if this were a dendritic cell, this is the link or the bridge between innate and adaptive immunity. Okay? So this is going to activate T cells, which are part of the adaptive immune system. All right, maybe that's the end of the movie. Let's see if it is. It really wasn't a very good movie. Yeah, I guess that's the end. Okay. And then you got to read the copyright because somebody worked hard on it, I guess. All right, yeah, so this is kind of what's going on here, right? If we have a bacteria that's phagocytosed, you got it in a phagosome, we get fusion of the phagosome with the lysosome, you get breakdown of the bacteria. This could also be a virus. And then the MHC, and what's not shown here is the MHC coming out, picking up some of these peptides and presenting it on the surface. So now T cells will know, if we have any T cells kind of floating by, remember I talked about lymph nodes or chat rooms for lymphocytes? So if this were to happen in a lymph node, sometimes what happens is macrophages will travel through the lymph, they'll be out on the surface getting these bacterial antigens here, and then they'll travel through the lymphatic system to the lymph node, which is kind of a chat room where T cells are, and then the T cell will be sitting in the lymph node, and they'll be chatting together and saying, hey, I picked up something foreign here, so get activated. And this is how the immune system knows that there's a foreign bacteria, that there's some foreign infection. Okay. Is that clear as mud? Good? Okay. That was a joke too. All right. So next. Yes. So how do macrophages and how do phagocytic cells recognize different types of pathogens? Okay, because not every we just said a bacteria or a virus. So but we know that there's lots of different viruses and there's lots of different bacteria. They have lots of different things on them. So there's things called toll-like receptors, and they're called toll-like receptors because does anybody work with any researcher who does Drosophila or fly research in here? Yeah, so the fly people are crazy. They have really weird names for things. But so the fly people named this, and then we figured out that it was also present on antigen-presenting cells, so that's why they're called toll-like receptors. Names some other weird stuff. So they have these structures on the surface that recognize foreign bits of foreign pathogens. So toll-like receptors, or TLRs, recognize what we call pathogen-associated molecular patterns, or PAMPs. Okay, so one of the things that you already know about is LPS, right? So where is LPS found? Well, yes, so lipopolysaccharide, but where is it found?
It's found on the cell wall,
So is it gram-positive or gram-negative? Gram-negative, right. So gram-negative cells have lots of lipopolysaccharide. Lipopolysaccharide is actually really toxic. So so sometimes, if you hear a physician talking about gram-negative sepsis, that means so septic means the person is sick and has circulating pathogen gram-negative bacteria, and the sepsis part kind of comes with the reason why they're really sick is because LPS is very toxic. It activates some cytokines that give you, anyway, it's a bad guy. So LPS can bind to something called TLR4. So TLRs are a family of proteins. So we have TLR1, which binds to acyl peptides that are produced by lots of different kinds of bacteria. TLR2 binds to peptidoglycan, okay, and some other things on mycobacteria, lipoproteins. What's peptidoglycan? The cell wall, right, absolutely. So it's a cell wall. So we have something right away on the surface of these cells that recognizes the cell wall. And remember, these phagocytic cells, so this is a phagocytic cell right here. Remember I said, as soon as the bacteria, those arms of as soon as the arms of the phagocytic cell contact the bacteria, so these arms actually have these toll-like receptors on them, these little receptors. As soon as it contacts the bacteria, it signals inside the cell. You don't have to worry about MyD88, but just know the concept that it signals inside the cell. And this says gene activation here. This is what causes the cytokine secretion. So remember, the cytokine secretion then goes down and it activates the endothelial cells on the blood vessel to make them have selectins, and the selectins then slow down to a roll the neutrophils that are rolling that are going through the blood vessel. So you're kind of putting everything together now. So we just kind of did a little bit of a deep dive. So these are the proteins on the surfaces of antigen-presenting cells or phagocytic cells that tell the antigen-presenting cell to make cytokines. Okay, and cytokines are ways to get more lipids. Cytokines and chemokines are ways to so that the immune system can call for help because there's a bacterial infection. Okay. So we talked about these on the cell surface, but there's also, so viruses can also infect cells, right? So there's also things inside the cell. We have toll-like receptors 7, 8, and 9. Those recognize DNA, viral DNA, viral RNA, so double-stranded RNA. We'll learn that viruses make double-stranded RNA. There's some that recognizes single-stranded RNA. So there are toll-like receptors on the insides of cells as well, just in case the cell gets infected with a virus. And antigen-presenting cells are actually pretty good at getting infected with a virus because they kind of want to be infected because they're going to be the cells that tells the immune system that something's wrong, something's infected. Okay. Next case. So fever. How many people have ever had a fever? Everyone's hands are going up. Okay. So I'm just trying to get classroom participation here, guys. So pyrogens are substances produced by microbes that cause fever. Okay? LPS, as I just kind of talked about, is one of them. And there are other toxins that bacteria produce that also cause fever. So there's exogenous things that cause fever that are exogenous, like from bacteria. And there's also endogenous factors that cause fever. Okay? So exogenous bacterial pyrogens induce the release of endogenous factors like interleukin-1, TNF, and IL-6. So I just introduced three terms that you're thinking, what the heck, Dr. Lake? So you just use this term interleukin on me. So interleukin-1 is a cytokine. Okay? And this is a cytokine, so it's called 1. It's one of the first ones that we discovered, right? Because we usually try to be logical, number things with 1 first. So interleukin-1 is produced by macrophages. So when a macrophage encounters a pathogenic microbe, it's going to produce interleukin-1. It's also going to produce something called tumor necrosis factor alpha. And so guess what? Tumor necrosis factor alpha wasn't discovered in a bacterial infectious disease setting. It was discovered in a cancer setting. And as a matter of fact, some cancers are very sensitive to tumor necrosis factor alpha. And when you put TNF on those tumors, the tumors go away. Or they regress. Okay, but both of these cause fever. IL-1 secreted by macrophages and dendritic cells, and TNF cause fever. And IL-6 also causes fever. Now these, especially IL-6, has other functions that actually helps B cells produce antibodies. So some of these cytokines have dual roles. And we'll be talking a little bit about cytokines later. Okay. So I said that we weren't going to talk too much about prostaglandins, but these endogenous pyrogens and exogenous bacterial pyrogens can also stimulate prostaglandin production, as you saw from the video. And prostaglandins then cause the hypothalamus to increase body temperature, which is why you get fever. Okay? So now you know. So now, when you're having dinner tonight, If the conversation isn't good, you can immediately talk about pyrogens and fever, and you'll be the hit of your dinner party. I know, every night.
As in you get hit.
Okay, so NK cells. I think I'm going to, because we don't get out until 1:30, right? So I do have a little bit of time. I will talk a little bit about NK cells, and then we'll come back to them later. So, because you do know what MHC is. MHC, the job of MHC is to present foreign peptides. So when I say foreign, it means non-self. Foreign peptides on it, so the immune system knows that the host is infected. Right? Everybody buys that? You're cool with that? Okay. So NK cells know that healthy cells express MHC class 1 on their surfaces. Sometimes tumor cells and cells that are infected with a virus say that I don't want the immune system to recognize me. I'm going to play tricks on the immune system. I'm going to suppress MHC expression on cells. So if a virus comes in, adenoviruses are particularly good at this. There's a lot of different adenoviruses. Some of them cause diarrhea. Some of them cause cold, like I think I have now. I don't know if it's adenovirus or not. Probably a rhinovirus. So sometimes MHC expression is suppressed on cells. So now the virus is kind of stealth, right? So the virus is infecting a cell, and if it has a mechanism where it suppresses MHC, there's no way for that cell to tell the immune system, to tell T cells, hey, I'm infected. Enter NK cells. NK cells are like men. NK cells are always on. Okay? That's a joke. Men are always on, right? So what happens is NK cells, let's see, where are we here? So here's a normal cell right here. Here's NK cells, and they have what's called an activating receptor. So if NK cells bind to the MHC, so let me just make it simple. If NK cells see MHC on the surface, then they shut off. But if they don't see MHC on the surface, then they suspect that something is wrong with that cell, and they kill the cell. They think that a virus is infecting the cell. Okay? So the inhibitory receptor here is not engaged, so this NK cell is going to kill this cell. So NK cells have to be inhibited. If they're not inhibited, so the inhibitory signal is dominant here over this activating signal. But as soon as the inhibitory signal, which is the MHC molecule, is gone, then the NK cell kills the cell because it figures it's virally infected or it's a tumor cell. Yeah.
Just looking at this picture, does it actually bind to both the inhibitory receptor and the activating receptor?
It does. But the inhibitory receptor is dominant. So that it says, don't do this because, I'm inhibited. But as soon as that MHC is gone, then it says do this. Yeah.
So basically this kills cancer cells. So would cancer be like it's producing too many cancer cells for the body to regulate?
Right. So cancer is a whole different topic. And not all cancer cells suppress MHC. So I don't really want to get into cancer right now, but I just did want to let you know that NK cells will kill tumor cells. Okay. All right, any other questions? All right, so we have a little bit of time left to do complement. Okay, so complement is a cascade of enzymes, of factors secreted by the liver. Okay, so you have lots of these individual proteins produced by the liver. It's like alphabet soup. I hate it. You'll hate it too. But I just want you to get the concept here, because I'm not going to test you on a lot of complement stuff. So you have this, just sit back and relax and listen. Then you can take notes as we go through it again. So you have this protein floating around. Everybody has this protein floating around that's made by the liver in your blood called C3. And C3 is called C3 tick over. C3 just constantly kind of ticks over and spontaneously forms, it splits into C3A and to C3B. So here we go with the alphabet soup. C3b associates with gram-negative cell walls. Okay? Sometimes gram-positive too. Gram-negative cell walls. Once this becomes C3b, so it splits, it just spontaneously splits in your blood, then it can associate with something called factor B. Okay? So factor B also has to split, and it does this by associating with factor D. Okay? So now we have factor D splitting factor B, so now we have C3b, big B, little b. Told you you're going to hate this. This is called a C5 convertase. Okay? The C5 convertase, C5 comes in here and gets split into C5a and C5b, and then this forms the genesis of what's going to be a pore. So as I mentioned previously in the lecture, the idea of complement is to punch holes in bacterial cell membranes. And or in bacterial cells, not just in their membranes, but it goes through the whole bacteria, the cell wall, and the membrane. So if you punch a hole in a bacterial cell, what do you think is going to happen?
It's going to die.
Right. So the good stuff's going to come out, bad stuff's going to go in, and the cell isn't going to be able to regulate its homeostasis. Okay, so it's not going to be able to survive. So we have C5b here attracts C6 and C7, and then C6 and C7, C5b attracts C8, and this whole complex here attracts lots of C9, and C9 forms the pore. Okay? So C9 is the thing that polymerizes here and forms the pore. All right, so the whole objective for complement is to punch holes in membranes. Okay? And unfortunately, you have to go through a big cascade in order to get there. But the reason we study this and we understand complement is some people have defects in complement, where they can't punch holes in the membrane. Some people are defective in C5. And if you're defective in C5, you're going to get certain infections, not all infections, you're going to get certain Neisseria infections. So you're more prone to meningitis, Neisseria meningitis, or Neisseria gonorrhea, for example. So Neisseria really can be killed by the complement cascade unless you're deficient in C5. So some people are deficient, some people actually overproduce something. Some people are deficient in the there's other molecules in here, not factor D, but there's other molecules that control complement deposition. Some people don't have those molecules, and they actually lyse their own cells. So complement will lyse their own cells, and that's bad. Okay, so this is called the alternative pathway of complement. We're starting with the alternative pathway because the classical pathway that I mentioned early on in the lecture, the classical pathway uses antibodies, and the antibodies are part of the adaptive immune response. Okay, so we're going to start with the alternative, and we start with C3 when we talk about the alternative pathway. Okay, I think we're near the bitter end here. Yeah, so here, this is what it looks like, right? Holes punched into bacterial cells. This is what I just talked about. And one thing I didn't mention is C3b is an opsonate. What's an opsonate? No takers? Yeah, I'm tired of being here too.
Does it have to do with coating the cells on these?
Absolutely. An opsonate is coating. It's like it coats the cells. We say something is opsonized, it's coated. Okay? Just a crazy immunologic term. Okay, so C3b can coat cells. Okay? And there's actually C3b receptors on phagocytic cells. So nothing goes to waste. And then remember, we're splitting things. Remember, we take C3 and we split it to C3b and C3a. We do the same with C5, C5b, and C5a. C3a and C5a are anaphylatoxins, which means, right, from protection. So they're actually not protected. So if you lyse a lot, if you activate a lot of complement, this triggers degranulation of phagocytic cells, and you can get a kind of a runaway inflammatory response. And you don't want that. And in some clinical cases, a lot of C3 and C5a are consumed, and you get kind of a runaway, and some autoimmune diseases as well, like lupus and sometimes RA. Okay, so the last thing is a clicker question, and since I'm going to give you a point for the clicker anyway, we could just do it by show of hands. So I'll let you read this and discuss it with your neighbor.
B.
Oh, this is easy. Okay, how many people say A? How many people say B? How many people say C? D? All right, so B must be the right answer. 200 grains is pretty good. Okay, so have a good spring break. Think about immunology.